Can somebody confirm you can hear me?
We can hear you.
Hello?
We can hear you.
Um, can somebody hear me? I'm not hearing anything.
Yeah, we can hear you.
Let's
I
see if
don't
my
think they can
volume
hear
is not
us.
working. All right. Ella, I think you're trying to say something. I didn't hear anything, but my volume wasn't on. So
Oh, okay, we can hear you.
great, great. All right. Before we begin, are there any questions? Not hearing anything. So, oops, get that out of here. Let me share my screen here. Oh, it's not right. All right. You should be able to see my screen now. Great, but I'm not sure. All right, today is November 19th, and we should be starting chapter 14. I think we need to finish chapter 8. Let me warn you, there's going to be a quiz this Wednesday, quiz 5. Quiz 5 is only going to cover chapter 8. Any questions about any of that? Now, for this entire week, both Tuesday and Thursday, there is no lab. You should be working on your unknown project. I will be logging in for the first 15 minutes of class to answer questions. But after, what is it, the last 15 minutes, meaning 6:45, I'm going to log out. So if you have questions, make sure you log in between 6:30 and 6:45. All right, any questions about any of this? If not, let me get to chapter 8. I was trying to do that. When I couldn't hear anyone, so I decided to, I think they've changed this now. I don't remember where the help is. I want to find something, and I don't remember where that is. Oh, there it is. Now we're further than that. So let me get rid of that. I think we're further than the operand model also. Is that correct? We've talked about the operon?
Yeah, we talked about the operon, and I think we left off on transduction in bacteria. I think the generalized
Yeah, I've
transduction.
got, we're right here. Is that what you think?
Yep,
All
that's
right,
what I
great.
have.
All right. I had too many two here's from earlier, and I need to remove them, otherwise I get confused. All right. So we talked about transduction and stated that viruses can move genetic material from one cell, the first cell the virus infects, to another cell, a recipient cell. And this only works when the virus makes a mistake and puts the genome of the first cell into the virus and then injects that genome, the DNA, not the entire genome, just a portion of the DNA, into the recipient cell. And there are three ways that DNA may spread around in a population: transduction being the first, conjugation being the second, and then the third being transformation. These happen to populations of bacteria cells. They don't happen very often to human cells. For one thing, our cells are not great at taking up DNA, and then it'd be very unlikely that your cells would ever contact with DNA from another human. I mean, it's just not likely. So transformation doesn't really happen in eukaryotic cells, at least human cells. Sexual or conjugation doesn't happen either, where DNA moves from one cell to another cell. Although we are a sexually reproducing species, in which case that is, well, it's sexual reproduction. And uh, in that sense, we have sexual reproduction. We do not have conjugation, where a plasmid moves out of one cell into another cell. Uh, Very unlikely that we have transduction, although transduction is theoretically possible. And in our nose cells, and throat cells, it probably happens all the time, where perhaps one person in their nose picks up the DNA from a virus from somebody else's cell. However, that only puts the DNA in that one nose cell, in which case, who cares when the patient then dies, that DNA is lost. It's only important transduction if it were to happen in a genome cell where somebody's either their egg or their sperm picks up a virus which inserts a gene from another cell into it. And that doesn't happen, at least not very often. I mean, it's theoretically possible, but not very often. And like I said, it does happen probably all the time in our nose and throat cells, but it doesn't do anything, at least to the population, meaning to the genome of the population. It has to happen to a germline cell. So in humans, this almost never happens, transduction. Any question about any of that? All right, let's talk a little bit more about plasmids. Plasmids are small circular pieces of DNA outside of the chromosome, why they're called extrachromosomal, that do contain genes. The difference between a plasmid in a bacteria and the chromosome is the size of the piece of DNA. A plasmid shown here has a few genes, I don't know, maybe 5 to 7 genes shown here. And a chromosome has thousands, maybe even hundreds of thousands of genes. So much larger piece of DNA. The plasmid can replicate independently of the cell's chromosome. And I actually already showed you that. In conjugation, we mentioned that this plasmid reproduces and sends one copy into the F minus cell. The chromosome of the F plus cell did not reproduce. So the two are reproducing independently of each other. Any question about any of that? Plasmids are very important in bacteria because a plasmid is much more likely to be exchanged between different bacteria cells than the chromosome would. Any question about any of that? All right. There are different categories of plasmids, and that's all you need to know. You don't need to know the names of them or what they do. Just need to know there's different types of plasmids in cells. And I should mention that there are some eukaryotes which contain plasmids. Yeast cells contain plasmids, or they may contain plasmids. I should say it that way, because a bacteria cell may contain a plasmid, but certainly not all of them do, and they don't have to contain a plasmid. The same is true of a yeast cell. It may contain a plasmid. Human cells do not contain plasmids, at least that I'm aware of, although we do have a extrachromosomal piece of DNA in the mitochondria and in the chloroplast, if you're a green plant cell. It's not really a plasmid, it's a separate molecule of DNA, and it's only in those locations in the cell. All right, let's talk about transposons and insertion sequences. Transposons are segments of DNA that can move from one region of DNA to another. And initially, I had real trouble with that because the genome was, when I was taught, was, I don't know, inviolate and unchangeable. And a transposon can move around in one cell to another location in the chromosome. In eukaryotes, a transposon can even move to another chromosome. It certainly can move around in the segment of DNA. And in bacteria, a transposon, if there's only one chromosome, it just moves from one part of the DNA to another part, and it can move into a plasmid or vice versa. Transposons always have an insertion sequence. That's a inverted repeat. Let me blow this up so you can see it better. They always have an inverted repeat on the ends and a transposase gene. The transposase gene actually will cut the region of the DNA that the transposon wants to move into and then anneal it. And I suppose it also anneals the part of the DNA, like, let's say this is a chromosome over here. It cuts out the transposon and then anneals the DNA back together. So that's what the transposase gene does. And it has these inverted repeats. It's thought that these inverted repeats, let me see if I can see ACT right there, ACT, an inverted repeat. And then also on this side, ATC, ACT, an inverted repeat. It's thought that these inverted repeats help this transposon insert itself into a new location in the genome. So the transposase gene cuts out the transposon, reseals the DNA, and then cuts open another piece of DNA, which the transposon then moves into. Complex transposon can carry other genes other than, I guess, the transposase gene. And a complex transposon usually has another gene in between a complete transposon over here and another complete transposon over here, meaning it has not just one insertion sequence, it actually has an insertion sequence there and there, there and there. And then two transposon genes, one there and then one there. Any question about any of that? Transposons are very important because they can move around a gene from one location to another, such as the kanamycin resistance gene. And then transposons are also important because they can move a transposon from the chromosome to a plasmid. And then the plasmid is much more likely to move into another bacteria cell. And in this case, it'd be much more likely to spread the kanamycin resistance gene. Any questions about any of that? So let's talk one slide about genes and evolution. Genetic variation can only result, meaning a new genetic variant in the population can only come about by mutation. We talked about an example, and that is over 10,000 years ago, humans only had brownish eye colors. And then 10,000 years ago, someplace in Northern Europe, we had a spontaneous mutation, and now humans contain a blue-eyed allele as well as a brownish-eyed allele. And that came from spontaneous mutation, creating genetic variation, at least in regard to eye color. But once that mutation then comes about, it can be resorted with crossing over and transposition, meaning it can move with another assortment of genes. Like initially, the blue-eyed gene was linked to the blonde hair gene, but because of largely crossing over and transformation, is that the word I'm looking for? No. Whatever word I'm looking for. um, no. Conjugation? No. I don't remember. Whatever crossing over, recombination, there it is. That's the word I'm looking for. Recombination. The blue-eyed gene got on, at least some people, the same chromosome as the brown-haired allele. And so nowadays those can sort. And then transposition can also move the gene from one location in the chromosome to even another chromosome. And so transposition and crossing over can make new genetic variations with other genes, meaning it's kind of shuffling the deck of cards. You still have the same deck, but they're going to sort differently, the different genes. And then once mutation creates the genetic variation, at least in bacteria, can move into other cells by either transformation, conjugation, and transduction. And as I mentioned, transformation, conjugation, and transduction are 10 times more likely than the genetic variation arising by spontaneous mutation. All of this genetic variation drives evolution via natural selection. And so that's how genes and variation and evolution occur. Any questions about any of that? All right. That's it for chapter 8. Any questions? If not, let's move on to our next chapter, which I think is chapter 13 or 14. I don't remember. Chapter 14. Okay, as is normal, I'm showing you the chapter name, chapter 14, Principles of Disease and Epidemiology, the major goals of this chapter, and a rough outline. So always know the terms. In this chapter, the terms are extremely important. If you simply know the terms of this chapter, you'll actually learn 50% of the material in this chapter. Any questions about any of that? And to know all of the terms and goals, just take a look at the chapter objectives. I've only got 3 major terms in this one: pathology, etiology, and microbial antagonism, but there's a bunch of other terms in this chapter. And then 2, understand the relationship between the normal microbiota and its host. 3, understand the stages of disease development, meaning disease, when it strikes, normally goes through certain stages of development. And then lastly, understand what nosocomial infections are and how to control them. Nosocomial infections are actually one of the most important topics that you can learn in microbiology, because it is an extremely important topic for those of you who go on to work in the allied health field. Any question about any of that? All right. Some terms: pathology is the study of disease. Etiology, the study of the cause of a disease. For example, the etiologic agent of the disease anthrax is the bacterium Bacillus anthracis. And that was discovered by Edward Koch, Dr. Koch, when he first proved the germ theory of disease. Any question about any of that? All right. Now remember, Dr. Koch did not propose the germ theory of disease. He was the first person to show that a germ could cause disease. All right. Pathogenesis is the development of a disease. Infection is the colonization of the body by pathogens or the presence of a microbe where the microbe is not normally found, such as E. coli is normally found in people's gut, but if it's found in the lungs, it can cause pneumonia because E. coli is not normally found in the lungs. In fact, E. coli is not normally found anywhere other than maybe on the skin and in the gut, the mouth, I guess. Disease is an abnormal state in which the body is not functioning normally. Some microbes can cause disease, but you should know that those microbes that can cause disease are pathogenic, and there's only a few of the microbes that are pathogenic, meaning can cause disease. Some microbes are even beneficial to the host, such as the microbes of the normal microbiota. All plants and animals have large communities of microbes found in association with them. And these microbes we call the normal microbiota, microbes that permanently or nearly permanently colonize a host. Now, let me tell you, when we're talking about the human normal microbiota, you should mention the human normal microbiota because it's different than another animal or plant's normal microbiota. For example, cats, domestic kitty cats, have Bartonella henselae as part of their normal microbiota, and it does not cause disease in cats. However, in humans, if the human gets Bartonella henselae, we call it cat scratch fever, meaning it does cause disease in humans. Any question about any of that? All right, the normal microbiota is only found in certain regions of the host, only certain regions of the body. Disruption of the normal microbiota and the host balance appears to be associated with disease and impaired immune function. For example, when you take oral antibiotics, you're going to decrease the microbes in your gut. And that can frequently lead to either diarrhea in the patient or constipation. And you can raise your hand if you want to indicate that that's ever happened to you. Certainly it's happened to me. It doesn't always happen, but occasionally it happens. And fortunately, the normal microbiota gut balance reestablishes, and then the diarrhea or the constipation ends, usually within a few days. Any question about any of that? All right, I mentioned the normal microbiota is only found in certain regions of the body. Let's talk a little bit about that. So we find the normal microbiota on the skin, on the mucosal membranes open to the outside of the body, meaning the eyes, the mouth, actually the ear, the urinary and reproductive systems, as well as the anus. Microbes are found in all of those regions, as well as the upper respiratory tract and the entire gastrointestinal tract. Any question about any of that? microbes, part of the normal microbiota, meaning are not normally found internal to the body, such as in the blood, cerebrospinal fluid, muscle, nervous tissue, and bone. If ever we have microbes growing in these regions, that would be an infection. For example, normally people do not have microbes, at least many, in our lungs. And if you have microbes in your lung, we call it pneumonia. But the upper respiratory tract is loaded with normal microbiota. Any question about any of that? All right, transient microbiota may be present for days, weeks, or months, but unlike the normal microbiota, when they colonize a host, they don't stay with the host for the host's lifespan or near their lifespan. For example, after I was born, I got Staphylococcus epidermidis, probably during my birth, certainly when I was touched by the doctors and my parents, and it has continued to be on my skin and continues to be there and probably will be there for my entire life. That would be a normal microbiota, something that colonized me after I was born and then remains with me for my entire life. And that's normally the way normal microbiota are. But once I had an example where I'd been testing myself routinely in microbiology. I'm just checking my skin to see if Staphylococcus aureus was ever on my skin. And for many years, never was. And I probably checked myself 3 times a year, sometimes 4 times a year if I was teaching in the summer. And it's just because I was in the lab and we were doing this experiment, and I gave the students MSA plate and said, okay, sample your skin, determine if you're one of the Americans who has Staphylococcus aureus on your skin. And we grew it up. We obviously taped those dishes shut because Staphylococcus aureus is a human pathogen. And for many years, I never had Staphylococcus aureus. And then I went and visited my aunt who had a Staphylococcus aureus infection of her skin. And she, when we went and visited her, insisted on hugging us. And after that event, when I sampled my skin, which was just a week or two after that, I had Staphylococcus aureus on my skin. And I said, geez, transient microbiota. Now I have Staphylococcus aureus. And I said, geez, I hope that doesn't last. And sure enough, 3 months later, in my next class and all future times I tested my skin, I never had Staphylococcus aureus show up in the lab. So I never had it any further on my skin. So it was a transient microbiota that I got from probably touching my aunt, hugging my aunt, and it only lasted for maybe a few weeks, and certainly only a few months, not longer than that. Any question about any of that? All right. The normal microbiota is about 4 times 10 to the 13th bacteria cells on a human. And that's important because humans only have about 3 times 10 to the 13th human body cells, meaning we have more bacteria cells than we have our own body cells. And that begs to ask the question, are we more microbe than we are human? And don't let that freak you out too much. Any question about any of that? All right, let's take a look at some human normal microbiota. There's the normal microbiota, the orange cocci, in the nasal epithelium. These are not rods. That's actually oh, I forgot what you call that. Villi of the nasal epithelia. And here we see some normal bacteria in the stomach, and their spirochetes, or maybe a curved rod right there and there. These are not normal microbiota. That's actually the villi of our stomach cells. And there's a stomach cell there. And then here we're looking at normal microbiota in our intestine. And there is a rod. That's sort of a bent rod. That's another bent rod. And then that's almost a cocci. And these are coccobacillus right there and there and there. This right there is not cells. That's actually food that's being digested. Same with that there. Any question about any of that? All right. The human normal microbiota has been found in some recent places that have surprised people. And this isn't so recent anymore, but it came out of studies done in 2014 where they found that the adult female bladder has normal microbiota in it. And it was assumed previously that the bladder was normally without a normal microbiota. And then if ever we had microbes in the bladder, it was a urinary tract infection of the bladder. But now we know that females, at least females, have a normal microbiota in their bladder. And it's always there, I suppose, after you're born. Sometime after you're born, you get it. I don't know if males have it, and they've never done studies on males to determine if they have normal microbiota in their bladder. The reason why males may not is they have a longer urethra, so it'd be harder for the normal microbiota to get up to the bladder. But at least females have normal microbiota in their bladder. And this is sort of interesting, and that is human breast tissue has normal microbiota in it, meaning mother's milk, even when it's inside the mother, is not sterile. It has normal microbiota in it. And this is actually important for the baby because the mother is giving the baby normal microbiota in her milk, as well as obviously milk. And this was a surprise to humans, and I guess I'm not sure why it was, because the nipple has to be open to allow the milk to come out. It's just people didn't realize that the normal microbiota can get inside the nipple and then inside the female breast tissue. Any questions about that? But once again, they've never tested male breast tissue, and so I don't know if males have normal microbiota in their breast, but obviously they don't make milk, so it's not terribly important. This one surprises me that people didn't realize that the human placenta has a unique normal microbiota. And the reason why it surprises me is, if you don't know, the female reproductive system is open to the outside. And that is why the females can have, a bacterial infection of their ovaries, because the microbe can get in the urethra, and let me see if I can get that right. Not the urethra, but go up the female reproductive system through the fallopian tubes, through the uterus, all the way to the ovaries. And normally that doesn't happen, but occasionally it does happen. And it surprises me they didn't realize that the placenta can have a normal microbiota, because the placenta is obviously open to the outside. But it was, and when they looked at the placenta, they realized that the normal microbiota found in the placenta is only found in the placenta, meaning humans have a normal microbiota that is only found in the placenta. And this is a little surprising, because the placenta is only in a pregnant female for a few months, and then it is like an after birth, so that it's discarded. And then there's no placenta in the female. And but while she is pregnant, she has a placenta and it has a normal microbiota, which is found no place else in the human body. I mean, some of the microbes are, but the unique microbiota of the placenta is only found in the placenta. Any questions about any of that? All right. The normal microbiota does establish a relationship with the host. The normal microbiota can, for example, create microbial antagonism between the normal microbiota and a bacteria that is a pathogen wanting to get established on a patient and then cause disease. So the normal microbiota or any bacteria can engage in competition with another species of bacteria, including a pathogenic species, where the normal microbiota can prevent the growth of pathogens for occupying the niche that the pathogen might want to occupy. For example, let's say I have a Staphylococcus aureus that falls on my skin. Well, my skin is covered with Staphylococcus epidermidis, as well as Micrococcus luteus, and some other normal microbiota. And if the Staphylococcus aureus falls on top of the normal microbiota bacteria, it's likely not to get established because the space is taken by my normal microbiota. And then the two can compete for resources. Like my normal microbiota, even if the Staphylococcus aureus falls on the skin where there's not a bacteria, it will be competing with the normal microbiota for the resources on the skin, meaning the food and resources the Staphylococcus aureus wants. And if the Staphylococcus aureus doesn't get enough, it's not going to grow and then cause disease. So they can compete for resources. The normal microbiota can also create acids, making the environment unfavorable for the pathogen to get established. This is especially true of the normal microbiota in our mouth and in our urethra and lower reproductive system, which for males would be the urethra, making the environment too acidic for many pathogens to get established. Furthermore, normal microbiota can produce a bacteriocin. This is a protein which is produced by our normal microbiota that inhibits the growth of similar bacteria. For example, we can have E. coli producing a bacteriocin, which then inhibits the growth of Salmonella and Shigella, which are two bacteria similar to E. coli. And then the bacteriocin will prevent the Salmonella and the Shigella from getting established and then causing disease in the patient. Any question about any of that? You can think of a bacteriocin as a very narrow acting antibiotic. It's even more narrow than the most narrow antibiotic, meaning the bacteriocin produced against E. coli only will prevent Salmonella and Shigella from working. They would not prevent other bacteria in the gut from growing, like Enterococcus faecalis. It would not inhibit that bacterium from growing, which is normally in our gut. Any questions about any of that? I've often wondered why we don't use bacteriocins when we have an infection, like a bacteriocin from E. coli, when we have a Salmonella or Shigella infection. And I did read an article, gosh, it's getting close to 5 years ago now, where they were experimenting with bacteriocins to treat patients against a bacterial infection. So at least it's been used as an experimental treatment, but it's not been routine. And it would be good because, like I said, if you got a salmonella infection, you take antibiotics to kill the salmonella, where you're also killing off other microbes in your gut. And if you were to use a bacteria syndrome E. coli, you would only inhibit salmonella and then a bacteria species very similar to salmonella. You would not hurt Enterococcus faecalis. That's the number one bacterium you find in your, well, I'm not sure if it's in the gut. It's certainly the one in your feces, the number one bacteria found in your feces. And then E. coli is also a bacterium that's very high in your feces, at least fresh feces. All right, any question about microbial antagonism? Competition between two bacterial species? We can use probiotics. This is the consumption of live microbes intended to exert a beneficial effect. For example, women taking yogurt containing live microbes are much less likely to get a fungal infection. And then humans taking bacterial containing yogurt, meaning a live culture yogurt, are much less likely to get a bacterial infection in both their urethra as well as their intestinal tract, because the microbes in the yogurt compete with the human pathogens. Any question about any of that? All right. The relationships between the normal microbiota and the host is one of symbiosis, where two different organisms or populations live together. The normal microbiota and the host live together in symbiosis, and it's a specific type of symbiosis, either mutualistic symbiosis or commensalistic symbiosis. In mutualism, both organisms benefit. For example, E. coli forms a mutualistic symbiosis with us, where E. coli gets a home and gets food that we give to the E. coli, but E. coli helps us because it helps us digest our food. Any question about any of that? For a cow, the reason why they can digest grass is they don't actually digest the grass. They have microbes in their gut which can digest the grass. And obviously, that cow would not be able to digest anything of the grass if the microbes didn't digest it first and break down the cellulose and then share that broken down food product with the cow. If they break it down to glucose, they can share glucose with the cow, or they can just break down the cellulose, and then the stomach of the cow can break down the grass further. Not sure how to word that because it isn't grass after you've broken down the cellulose, but you got my point. The other way a normal microbiota can be is a commensalistic symbiotic relationship. Commensalism is where one organism benefits and the other is unaffected. An example of commensalistic microbe in humans is all of us have one nematode, at least on average, in each eye, and only one. And this nematode does not damage the humans, does not eat the human eyes. I'm assuming what it's eating is the microbes that fall into the eye, because it's not eating the eyes. And this microbe is a nematode, but it's microscopic. You can't see it. The point is the human gives the microbe a space to live. And sometimes you can give the microbe something to eat, although humans don't give the nematode something to eat. But another normal microbiota, we could give something for the microbe to eat. But the microbe doesn't do anything for us. It doesn't cause disease. It doesn't benefit us. And the nematode, we don't get any benefit out of it, but the nematode gets someplace to live. So that's a mutualistic, not a mutualistic, a mensualistic symbiotic relationship. Any question about any of that? A parasitism is where one organism benefits at the expense of the other, meaning one organism benefits, the other organism is harmed. All microbes that cause disease are a parasitic, symbiotic relationship. So all human pathogens follow parasitism, where one organism benefits, it gets a home, it usually gets food from us by killing our cells, and then the human would be damaged by getting a disease. Any question about any of that? All right, the normal microbiota does not normally cause disease, but they can cause disease in certain circumstances. And this disease is caused an opportunistic microorganism where some normal microbiota can become opportunistic and cause disease under certain circumstances. Normal microbiota do not normally cause disease in a healthy individual, but under certain conditions they may, such as the individual gets immunosuppressed by therapy or treatment, or some other reason becomes immunocompromised, the normal microbiota may grow in regions where it shouldn't. Such as in individuals with compromised immune system, a patient receiving chemotherapy for cancer treatment, or an AIDS patient. And then the normal microbiota can usually grow to levels it should not be, as well as grow where it should not be. Like if somebody gets E. coli in their ear, that would cause an ear infection. E. coli in the normally found in the gut, but if it's found in the lungs or the ear, it's causing an infection, an ear infection or pneumonia if it's in the lungs. Staphylococcus aureus is found in about 50% of humans in the United States, nose and throat. If you look at the skin, only about 20% of Americans have Staphylococcus aureus on their skin. But Staphylococcus aureus is a human pathogen, and individuals who have it on their skin are much more likely to get a boil, a skin infection, than individuals who lack Staphylococcus aureus on their skin. Any question about any of that? All right, we've talked about pathogens causing infectious disease, and we also talked about how Dr. Koch proved that Bacillus anthracis causes the disease anthrax. He did that by following Koch's postulates, the steps he used to determine that Bacillus anthracis caused disease. And those are so important in microbiology that we call them Koch's postulates, the steps you use, or Dr. Koch used, to show that a given bacteria causes a given disease. First, you start with a bunny or individual or patient which is sick, and then you isolate out of that individual or bunny microbes. You grow up these different microbes as pure cultures, meaning you streak them out for colony isolation 2 times and then grow it up, and then you got a stock culture, and then you inject some of that stock culture, meaning a pure species, into a healthy bunny. If that healthy bunny later becomes sick with the same disease as the first bunny, then we say that that microbe that we injected into the patient or bunny is likely causing that disease. But Dr. Cox said you need to go one step further. You can't just show that this microbe causes this disease. You then have to isolate from the sick animal the disease-carrying organism and then grow it up. If you can do all that, that's Cox postulates. And these steps Dr. Cox did do, showing that Bacillus anthracis causes anthrax. And he did show that if he injected Bacillus anthrax into mice, that it would cause the disease anthrax in the mice. Any question about any of that? Let me mention the reason why the bunny is on its side is because it is sick here as well as there. And then if the bunny is healthy, it's standing up. Any questions? All right, we mentioned the Cox postulates are used to show that a given microbe causes a given disease. But there are some exceptions to Cox postulate. Like, what do you do with microbes that have unique requirements and cannot be grown in tissue culture? If it can't be grown in tissue culture, you can't perform Cox postulates. And then what do you do if multiple organisms can cause the same disease, signs, and symptoms? For example, when many different microbes get in the lung and cause disease in the lungs, we call them pneumonia. But there are many different species which can cause pneumonia. And if you have many organisms causing the same disease, you can't use Cox postulate to then prove it. And then what do you do if there's moral and ethical conditions for a human-only disease? For example, we believe that HIV causes the human disease of AIDS, but we've never injected HIV into patients to determine if those patients later develop the disease AIDS. So how do we know that HIV causes the disease AIDS? Anyone have a guess? Now, how we know is we've done vast epidemiological studies where we study a large number of people, and then we watch these people over time, meaning you're assaying over time. And what they did was they found some of the people who are HIV negative on their second or later tries, discovered the patients had by their own means become HIV positive. And then they followed those people for a length of time, and they found that they eventually developed AIDS. And that was done actually earlier before there was all these drugs for treating AIDS. Any question about any of that? Now, the studying large populations in an epidemiological study for something like determining if the disease AIDS is caused by the virus HIV. We have done that, but I lost my train of thought. What the heck was I trying to think of? So we followed people over time, and by their own, they became HIV. And then later, eventually, almost all of them developed into AIDS. Nowadays, that isn't the case because people can take medication and prevent AIDS from developing. And the medication is actually so good that these immunodeficient patients can be kept alive close to their normal lifespan. I mean, they're not dying in their 20s, and usually they only got infected, well, about age 18, but I suppose age 16 be the youngest. And obviously, you can get HIV younger than that, but it's rare. And if you do get HIV, I'm not saying to do this, but you can take medication and then live close to the normal lifespan. It's a little shorter than the normal lifespan, maybe 5 to 10 years shorter, but it's close to the normal lifespan. Which does anyone know what the average American's lifespan is now? I'm sure it's in the 80s. It might even be in the upper 80s or the low 90s, at least for women. All right. Some more terms for classifying an infectious disease. You can have a symptom, and that's a change in the body function that is experienced by a person as a result of a disease, but it is a subjective change, and that change is not apparent to an observer. An example of a symptom is when you have a headache. You know you have a headache, but nobody else can measure the headache to determine how bad it is. Any question about any of that? So a symptom is one that the patient knows they have, but nobody else can measure it. So nobody else really knows that the patient has it. A sign is a change in the body that can be measured or observed as a result of a disease. So the sign is a disease which the cells can be measured but first became infected. So for example, the bacterium Lithuania, no, that's not right. The bacterium which causes the infection can be measured, and then the symptoms cannot, but the sign could be. So a good example of a sign is when a patient gets a temperature. Everyone can measure that temperature by sticking a thermometer in the patient. So a sign is a change that the body, the patient, can identify, meaning knows they have, and then everybody else knows they have because they can measure it, such as they can put a thermometer in the patient and then get the temperature. So that would be a sign. A syndrome is just a specific group of signs and symptoms that characterize a disease. For example, you can talk about the AIDS syndrome, where they have certain signs and they have certain signs and symptoms. Any question about any of that? Such as the signs and symptoms characteristic of the AIDS syndrome. Few more terms: communicable disease, any disease that is spread from one host to another. So an example of communicable disease is measles, tuberculosis, and chickenpox. A contagious disease is a disease that is easily spread from one patient to another. Can anyone tell me a contagious disease? Come on, I know you guys know.
The flu.
You should know at least two. Yeah, the flu is a contagious disease, one that easily spreads from one patient to another. And then what are we in as far as a pandemic or epidemic right now?
COVID.
Mm-hmm. COVID-19 is also a contagious disease. And which of these three diseases is communicable but is not contagious? Meaning it can spread, but it doesn't spread easily. Measles, TB, or chickenpox?
Chickenpox?
No,
I'm
measles.
I'm saying again?
Measles?
No, measles is very contagious. That's one of the few diseases which is much more contagious than COVID-19, for example. Chickenpox, you guys probably don't ever have chickenpox parties. When I was a boy, when somebody got the chickenpox, they would invite children from their neighborhood or their friends over who were healthy to expose them to chickenpox because you wanted the child to get chickenpox when they were healthy. You didn't want them to get chickenpox when they were sick. So we had chickenpox parties. So you obviously know chickenpox is a disease that spreads pretty easily, right? The one that doesn't spread very easily is tuberculosis. I was surprised that you guys didn't mention that because obviously you guys are worried about tuberculosis because you work in the allied health field, or you will, and so you're exposed to tuberculosis. And the good news is you're not as likely to get it. You can get it, but you're not as likely to get it as many other diseases because tuberculosis does not spread easily. Usually you need long-term and consistent exposure before you'll pick up tuberculosis. So tuberculosis is communicable, but it is not contagious. Measles is communicable and contagious. Any question about that? And another way of thinking about this is all contagious diseases are also communicable, but not all communicable diseases are contagious. And then we have some diseases which cause a non-communicable disease, a disease which is not spread usually from one host to another. For example, tetanus. And what's another one I was thinking of? I just lost it. Oh, no, that's not a good one. Botulism. There we go. I knew it was something related to tetanus. These two diseases do not normally spread from one patient to another. So when a patient gets tetanus or botulism, they're not going to spread it to another patient normally. There is one exception. Can anyone think of an example where you can have a patient who has tetanus and then it spreads to another human? Can you think of an example? Anyone? Well, you work in the healthcare facility, so I was going to say you can get tetanus from a needle prick. So when you handle your needles on a patient, you should dispose of that needle carefully and appropriately. And obviously don't stick yourself with the needle because you could get tetanus from a needle. Not terribly likely, but you could. And then the other way you could spread tetanus is if the patient has tetanus and then a cannibal comes and eats the patient. That cannibal could get tetanus, especially if they eat the patient raw or don't cook them well. Any question about any of that?
Yeah, I actually have a question about TB.
Uh-huh.
So there is such a thing as latent TB, and does it mean that person been exposed to TB virus, but it's not like active?
Um, TB is a bacterium, not a virus.
Sorry,
And when
bacteria.
a patient gets TB, initially they'll have an active infection, but then can become a latent, and I don't use that term with TB because what latent is one that does not spread, and I guess while it is latent, TB is not likely to spread. What happens is the host immune system mounts a response against TB and then surrounds the TB in a nodule. And so the bacterium cannot get out of the nodule and then spread to another patient. But the virus can break the nodule and then become an active infection, in which case the bacterium can spread to another patient. So that's
But
what we're talking about.
yes, but while it's sitting in a bubble, it can be
It's not in a
transferred.
bubble, it's a nodule where the immune
Nodule.
systems surround the microbacteria, a nodule.
So it can be passed to another person, right? While it's not.
While it's a latent, meaning it's inside the nodule, it's not likely to be passed from one patient to another. In fact, while it's all in the nodule, it would not be passed. But obviously, there could be some, while it is in the nodule, which might be passed, but not very likely because there's not very many. What's most likely to be passed when you have an active infection, and that's when the patient is first infected before the nodule forms, before the host immune response responds to the disease, or when the bacteria breaks out of the nodule. It can do that where, well, we'll probably talk, well, maybe we're not going to talk about that in this class. Yeah, we're not going to talk about that. Where the host immune system establishes a stalemate and then keeps the TB surrounded by a nodule, and that prevents the TB from spreading to other parts of the patient as well as spreading to other patients.
Okay, thank you so much. Very interesting.
The problem with TB is when you get older, your immune system weakens, and then the TB is much more likely to break the nodule and then get out and cause an active infection.
Thank you.
All right. Let's talk a little bit about occurrence of a disease. To understand the full scope of a disease, we need to know some details about its occurrence. The incidence is the fraction of a population that contracts a disease during a specific time period. The incidence is only measuring the new cases of disease. On the other hand, we have prevalence, and that's the fraction of a population having a specific disease at a given time. So that would be both the new patients and the old patients equal the prevalence of the population affected by disease. For example, in AIDS in 2007, the incidence, meaning the new cases in 2007, were 56,300 in all of the United States. However, the prevalence of patients with the disease in 2007 was 1,185,000. Obviously, the prevalence is larger than the incidence because the incidence only includes the new cases. Some diseases, we talk about their incidence, like usually when we're talking about the flu during a given time, like how many cases of flu there are now, we're talking about the incidence of the flu, which at the moment, I believe the incidence is still very low, but it's climbing because it's late in the fall. And it'll get highest probably in the winter. And then it'll start decreasing in the spring. And then there are some diseases like COVID-19 where we usually talk about the disease with the prevalence, meaning the total number of people who've come down with COVID-19 and gotten sick, which for the American population is now over 50%. It's probably over 65% now of the population who have had COVID-19. Any question about any of that? All right. when we're talking about the occurrence of disease, there's a few terms you need to know. There's a sporadic disease. That's a disease that occurs only occasionally in a population, like typhoid fever. It shows up in the United States, but not very often, so many people call it a sporadic disease. And then there's an epidemic. This is a disease that is constantly present. Excuse me, endemic. I didn't say that right. Endemic, a disease that is constantly present in the population. Like the common cold is endemic in the human population. And malaria is endemic to the tropics and subtropics of the world. Especially you find malaria in the tropics because the mosquito is around that can spread it, as well as the parasite. An epidemic is a disease acquired by many hosts in a given short period of time. An epidemic is usually when we have an increase in the number, and the influenza can sometimes in the wintertime cause an epidemic where the disease acquired by many hosts happens at a short period of time. A pandemic is when the epidemic becomes worldwide. So influenza and AIDS, as well as COVID-19, are now pandemic. They're all worldwide. But initially, COVID-19 was only found in China, in which case it was an epidemic in China, and until it spread out of China, it was only an epidemic in China. It soon became worldwide, becoming a pandemic. Any question about any of that? Another useful way of defining the scope of disease is to talk about it in terms of its severity or duration. An acute disease is a disease where the symptoms develop rapidly, the disease is short-lived, and then the disease goes away. A classic acute disease is the human cold. You get infected, you shortly develop a disease. The disease infecting agent becomes large in number in a very short time, and then the human cold decreases and goes away. There are many diseases which follow the acute disease duration or severity. Influenza is one, a little more severe than the human cold, but influenza is an acute disease. Hepatitis B, an acute disease, usually an acute disease, I should say. A chronic disease is a disease that develops slowly, persists for long periods of time, and usually increases in number over time. An example of chronic disease is hepatitis B, causing cirrhosis of the liver, and then the patient never gets rid of hepatitis B, and it becomes a chronic disease. With each incidence, it does more and more damage to the liver, which can cause eventual liver failure or maybe liver cancer. Any questions about acute and chronic diseases? A chronic disease does not have to be caused by a microbe. Tennis elbow is a chronic disease which develops slowly, persists for long periods of time, and usually gets worse with time. The more you play tennis, the worse your tennis elbow becomes. Any question about any of that? We do have diseases in between an acute and a chronic disease. You don't need to know the term or the example, but I'll mention the term at least. A subacute disease, having symptoms in between that of an acute disease and chronic. Subacute sclerosing panencephalitis is an example, and I'm sure most of you don't know subacute sclerosing panencephalitis So you don't need to know it. It's just one that doesn't occur rapidly and then be short-lived and go away. It generally starts fairly slow, persists for long periods of time, but not as long as a chronic disease. And then it does usually go away. Whereas a chronic disease not always goes away. For mononucleosis, it's a chronic disease, starts slowly, gets worse with time, persists for a long period of time, but mononucleosis usually goes away. Hepatitis B, when it's a chronic disease, the patient may live with hepatitis B for the rest of their life. That isn't true for all cases of hepatitis B, but it's true for at least some, if not many. Any question about any of that? All right, let's talk about the extent of host involvement. Infections can also be classified according to the extent to which the host body is affected. You can have a local infection, and that's where the pathogen is limited to a small area of the body, such as a skin infection caused by Staphylococcus aureus on the skin, causing a boil or an abscess. And as long as it doesn't go further, that's all you have is a boil. Eventually, the boil will heal and go away, and that's a local infection. However, you can have a systemic infection where the infection spreads throughout the body. Measles isn't a case where once the patient becomes infected, the virus spreads throughout the body, causing a systemic infection. That's why the measles patients usually get very sick, because it's growing in many places, many cells in the body, causing damage and making the patient sick. A focal infection is a local infection that then spreads to a systemic infection, like Staphylococcus aureus, causing skin disease, causing a boil. If that boil gets worse, the boil can grow until it reaches the blood vessel, and then the Staphylococcus aureus can be spread by the blood to other regions of the body, causing now a systemic Staphylococcus infection. It started as a focal infection and then, or systemic infection that then became, as a local infection that then became systemic, meaning the boil grew until it spread the Staphylococcus aureus into the bloodstream. Any question about any of that? All right. Some discussions of this extent of host involvement include bacteremia. That's the presence of bacteria in the blood, shown here and here, and septicemia, that is actually growth of bacteria in the blood. Usually clinicians do not look for septicemia. They'll just look to see if there's bacteria in the blood, and so they'll use the term bacteremia. We found bacteria in your blood. But if you have a specific test, you can test for the growth of bacteria in the blood, and then we call it septicemia. Toxemia is the presence of toxins in the blood. Viremia, the presence of viruses in the blood. Like chickenpox, the virus can go into the bloodstream, and you'd have viremia. Any questions about any of that? All right, if there's no questions, I'm going to stop here, and I'll see you next time in the lecture. If you have any questions, you can show up in the lab at 6:30. Remember, I'll only be there from 6:30 to 6:45 to answer questions. All right, goodbye.